Two more factors that affect the rate of enzyme-catalyzed reactions are the concentration of enzymes in the medium and the concentration of substrates. And we're going to look at these both in this video. We're going to start with the effect of the concentration of enzymes on enzyme-catalyzed reactions, and then we're going to talk about the effect of substrate concentration on the rate of enzyme-catalyzed reactions. So to first discuss enzyme concentration, we can have three scenarios in a specific medium. We can have a low enzyme concentration with respect to the amount of substrate, which means that the substrate is present in excess and there are not enough enzymes to be working on all of the substrates at the same time. Some substrates will have to sit and wait until the enzymes are done with the substrates they're working on to go work on them. This case is where we have excess substrates. And in this case, if you add more enzymes, then the reaction will start to occur faster, simply because the substrates that were sitting and waiting and not being acted on will be acted on while the present enzymes are working on other substrates. So the amount of substrates that is sitting in excess can now be worked on upon adding more and more enzymes. So in this scenario, increasing the concentration of enzymes in the medium increases the rate of the reaction until you reach a specific level where you added so many enzymes that you are now working at full capacity on all of the substrates. So adding any more enzymes is not going to affect the rate of the reactions because these enzymes are now in excess. They're sitting with no substrates to work on because all of the substrates are being worked on. So to repeat, if you have low enzymes with respect to substrate, if the substrate is in excess, the reaction is going to be slower than it possibly can be. If you have a balanced amount of substrates to enzymes, if you have an intermediate enzyme concentration, then you will have the maximum speed at which the reaction can occur because all of the substrates are being worked on at the same time. And beyond this point, if you keep adding more and more enzymes, these enzymes are going to be sitting, waiting, with nothing to act on because all of the substrates are being acted on. So now the enzyme is in excess, its active sites are empty, it's just sitting there, and here you are at a high enzyme concentration. Now the last variable which affects the rates of enzyme-catalyzed reactions is substrate concentration. Substrate concentration is very similar to what we just discussed in terms of enzyme concentration. If you have a low substrate concentration, now the enzymes are in excess. So this is just like when we added too many enzymes last time. Now we are starting with already too many enzymes. The reaction is going to be slow here. And you can increase this reaction. It has potential to be increased. You can increase its rate by adding more substrates. If you add more substrates, then the enzymes that are in excess, that are sitting there with their empty active sites, can now be acting on these extra substrates that you are adding. So you can increase the rate of this reaction linearly, proportionally, as you add more and more substrates, until you reach what we call Vmax, or maximum velocity, which is when all of the enzymes have substrates to work on. You reached the perfect balance of enzyme and substrate concentration. Beyond this point, if you add more substrate, then you are not going to increase the rate of the reaction because the enzyme's active sites are full. They're already working at maximum capacity. So if you give them more substrates to work on, they can't do anything. They're already working on substrates. So this is where we reach the plateau in the curve, in the graph. Here, the more you add substrates, you will not increase the rate of the reaction because you are already at high substrate concentration. The substrate is in excess. So you can see how similar these ideas are of substrate concentration and enzyme concentration. There is a balance of substrates to enzymes at which you can achieve maximum velocity, maximum efficiency of the enzyme-catalyzed reaction. And this concludes all of the variables that affect the rate of enzyme-catalyzed reactions and how we can manipulate them.